So our first company of the evening and speaker of the evening is Jim West, and he is the founder and CEO of a startup called Clara Biotech. One of the things, while Jim gets his slides up, one of the things that you're gonna notice tonight, we do always like to hear about the science and the medicine surrounding these companies. Well, when you go into biotech, you're gonna notice that a lot of these are very science heavy. So I'm going to say you're welcome in advance. And I know that this is going to generate a lot of questions from our audience. So Jim, go ahead and take it away.
Thank you, Katie. I'm Jim West, CEO of Clara Biotech. And we founded our company in 2018 to solve some of the major challenges and issues in the field of exosomes. And the goal of our company is to help move exosome technologies from the bench to the bedside. And this really represents an exciting and new opportunity to lay the groundwork for the next foundation of biotechnology. And let me start by telling you what an exosome is if you haven't heard of them before. Exosomes are the FedEx communication system of the body, and exosomes communicate and transmit cellular information like DNA, RNA, and proteins. They go between cells, and here's an image of one. There are about 100 nanometers in diameter, and there's about 1,000 exosomes to every cell in your body. They're prolific, and they have a number of really exciting and interesting applications in the field, starting now with delivery of RNA and nucleic acids to specific locations using the exosomes as a targeted delivery vehicle, but natural. Also, there's exciting applications in cancer immunotherapy where you can get the same benefits of CAR T-cell processes, but in an allogeneic platform where you can mass produce this product as a standard pharmaceutical. And a few years ago, we discovered that exosomes that come from stem cells inherit many of the same benefits and properties, including acting as an anti-inflammatory and promoting regeneration of tissues, but in an acellular platform. It also has exciting applications in the fields of biomarker identification, so liquid biopsy. Right up there next to circulating tumor cells and cell-free DNA, you have the ability to identify diseases like cancer, Alzheimer's disease, and other neurological conditions simply from body fluid samples. And the big challenge today is that most of the competing systems are based on some form of bulk isolation or density isolation that gives you a final product that is noisy with contaminants you don't want and can't get rid of. At Clara Biotech, what we've developed is a pure, active capture and release technology that gets exosomes and nothing else. And here's how it works. We start with an immunomagnetic bead, and to the surface we put targeted antibodies. These are totally customizable and tunable. We can put whatever combination we want on the surface, and it allows us to capture specific types of exosomes, tuning it to that specific need. Once it's bound to the surface, we can wash away all the non-exosomal content, and then we have a proprietary photo-release process that detaches the exosomes from the bead. And here's how it works. It's really simple. It works in any laboratory setting. It's based on immunomagnetic bead technology, which has been around for more than 40 years. So everybody knows how to put this in their workflow. You start off by mixing your sample, your body plasma, your urine, saliva, or cell culture media with our proprietary reagent. This is a single-use consumable. And once the exosomes are bound to the surface, you put it into the box you see here on column 2, and it releases the exosomes over a 20-minute period. The great thing about this platform is not only is it scalable and usable in any laboratory, but these exosomes are suitable for any downstream application, which is a really unique benefit in this field. Here's a comparison showing our technology to the gold standard, in this case, ultracentrifugation. With what you see here is on the left is a Clara bead with exosomes bound to the surface shown with the red arrows. And on the right is a solution of exosomes through ultracentrifugation, which shows a lot of noise and contamination. And what's really important is the graphs on the bottom. These are called Nanotracking Analysis Reports, or NTA. And what you see on the x-axis is a size distribution from 0 to 300 nanometers. And on the y-axis, you have quantity. And so on the left, with Clara's product, you see a narrow band of exosomes captured. And on the right, you get a much more noisy sample representing a lot of non-exosomal content. We can get highly functional exosomes too. And here you can see an assay using exosomes that have been taken up into dendritic monocytes. We fluorescently tag them, and you can see the signal that goes into this cell showing the green fluorescence. And we get highly functional exosomes relative to the gold standard. And this is really important to help solve future problems in manufacturing and scalable therapeutic grade exosomes. Like I said, we've been around since 2018. We started with an SBIR phase 1 from the National Cancer Institute. It's very competitive and very hard to get. We also completed what they call I-Corps at NIH, or do customer market validation at the very beginning. Shortly after that, we raised our pre-seed round to help get our exosome isolation lab service up and running and to move us forward into product development, which we're in the middle of today. In 2020, we started heavy development of our exosome isolation kit and also participated in Mass Challenge Boston, where we were able to connect to the Boston ecosystem. From that, we were able to get a number of great investors and advisors from the community. And today, we are more than 3/4 of the way through our seed round, which we're expecting to close in the coming weeks. And with all this in place, what we're doing right now is we're getting our first research use only exosome isolation kit to market. You can see it on the upper right of the screen. And this kit is something that we've got more than 10 beta sites around the world today using. And because of the goal of the company moving exosomes from the bench to the bedside, where we're going beyond the research kit is in supporting diagnostic applications for liquid biopsy and also solving serious problems around therapeutic manufacturing at scale. I founded the company 3 years ago. My background is in biomedical engineering. I also have a master's in entrepreneurship, and in 2016, I got one of the first mobile medical devices cleared by the FDA. My co-founder, Dr. Mei He, is the inventor of our platform. She's been developing this since 2012 and is an active researcher who's been published in Nature and other great journals. She's an expert in microfluidics, exosomes, and drug delivery. We also have Dr. Tom Kroll, a pharmaceutical drug developer who's helping us with pharmaceutical development and deal flow. Today, we have 5 full-time employees, 4 part-time employees, and we're actively sourcing for 3 new positions. Do you have any questions?
Jim, thank you. I invited our audience, but I will once again go ahead and put any questions that you have for Jim and Clara Biotech into the Q&A function. I'm going to start with a couple of my own, though. Again, I mentioned this at the beginning, right? This is pretty science heavy. For those of us that have been out of medical school for a little while, I loved the fact, by the way, that you reviewed what an exosome is for us, because many of us probably are thinking to ourselves, what is that again? But I'm guessing you talked about the use of this technology in therapeutics, and I'm guessing that purity is a really big, important piece of that. So talk about how your purity varies from the ultracentrifugation that you talked about.
Yeah, thank you, Katie. So the reason purity is so important and the challenges that the industry experiences with it today is when you do ultracentrifugation, you're effectively isolating based on a density gradient, or you're running it through a size filter and you're filtering based on size. And in both of these methods, we're talking about material that's 100 nanometers in diameter. And when you isolate this way, you end up with a lot of non-exosomal material. A lot of other stuff exists at this domain: cell debris, cell-free DNA, viruses, lots of stuff you don't really want. And so when you're looking at signals, you get a lower signal. You're looking for a needle in a haystack, and it gets masked. And when you talk about therapeutics, what the FDA and other regulatory bodies look for is pure, potent product. They want a clean, well-defined, active pharmaceutical ingredient. And today, with these filtration methods, it's very difficult to be able to cross that gap. And with our selective capture and release, being able to provide a pure qualified active ingredient, we believe we can overcome those hurdles.
Which is why I'm guessing you're starting in the research market. Clearly, there's a need there for this type of technology.
The whole field's only 10 years old, so it's a very, very young field. And I don't have a slide for it, but last year in 2021, there were 5 $1 billion licensing deals between pharma companies and exosome manufacturers partnering to get RNAs and gene therapies delivered to specific locations. And so it's really heating up. It's a very active field. You are right. And from a company development perspective, you don't want to go straight to regulated manufacturing. You want to build up your way there. So we're starting in the research space, and we're going to grow that organically.
Fantastic. So Dr. Macaluso is in our audience tonight, and he has my second question right on the tip of his tongue as well. He puts it a slightly different way, but please discuss the major clinical application that you are looking toward. And I would say, tell us a little bit more about the most recent use cases. What are they using the technology for?
Yeah, thank you, Dr. Macaluso. So the use cases, as I mentioned, obviously delivery of drugs like a liposome or a lipid nanoparticle. That's coming, and there's products moving through the pipeline for those activities. What I get really excited for, though, are some of those other things I kind of mentioned. So cancer immunotherapy. I mean, it's huge that you can have a product that has the ability to train your immune system how to fight diseases, but without requiring like CAR T cell. One of the challenges there is you have a little bioreactor for each patient. So you give your own material, and then 2 weeks later you turn it around into a therapeutic. It's very difficult to scale that in the traditional biotech world. With our platform and with what we can provide, you can actually create a large-scale manufacturing process for that. I also think that it opens up the door to regenerative medicine in a traditional way. So right now, a lot of that technology exists outside of the FDA, and it works off-label. And I think that we have the opportunity to create actual labeled drugs for regenerative medicine.
Very cool. So another question from our audience. Talk a little bit about, you mentioned pharma companies, but talk a little bit about the main strategics that might embrace this type of biotech.
Yeah, and I think I didn't quite answer Dr. Macaluso's question there as well. So the strategics that we're looking for, it's really, we are a platform. We're building pickaxes and we're selling them to miners. We don't know who's going to hit gold, but they'll be using our pickaxe when they find it. And so we're really looking to be a catalyst for the industry. We want to support everybody in the work that they're developing. And so there's lots of opportunity and lots of applications that are being developed, and we want to work with and support them. And so today, we are working with a number of researchers, diagnostic and therapeutic folk. To be honest, our scale, we're still early. We haven't got our first product out. So we're still a ways away from therapeutic scale activities. But I can tell you that on the diagnostic side, we have interested companies in looking at biomarker analyses. And we also have some research use only applications with some major pharma companies looking at actually supporting clinical trials by looking at pharmacokinetic markers and other things like that exosomes can provide in a non-clinical setting, but allows us to actually have a scalable platform before we go to commercial clinical uses.
Very cool. A couple more questions from our audience. Is this technology comparable to linker technology, and does it have the ability to cross the blood-brain barrier? That's a great question.
Yeah, so the short answer is yes. You can modify and load an exosome. So you can program it for different locations, and they do cross the blood-brain barrier. And so there's a number of really interesting applications looking at delivering CNS products to the brain and to specific locations.
Awesome. And one final question from another physician in our audience tonight. Tell us a little bit about your IP. How protected do you feel about your kind of secret sauce, your reagent box in regard to competitors?
Yeah, it's a great question. Again, this field, the immunomagnetic bead core technology has been around for 40 years. What Clara has really innovated is two things that we have IP around. One is we have a custom bead that's actually designed for exosomes, and it actually is able to capture a lot more exosomes to the surface based on that geometry. The second aspect is that release phase. And so it's very difficult to get these exosomes off. And so we have IP around that release phase. And then there's a third part, which I won't really go into here, but it's around downstream microfluidics that allow us to do automated processing in the future.
Great. Jim, thank you so much for being here tonight. Thank you for telling us all about Clara Biotech. This is some really exciting technology. And as you mentioned, I think at the forefront of what's happening in healthcare.
Can I say one last thing on the poll, Katie?
Sure.
I would just like to say, I'm fortunate enough to be in the room right now with a lot of smart, connected people. And, most of you might not be super familiar with exosomes yet, but you probably know people that are in areas of development and research that this technology could play a part in. And what we're really looking for are high-level qualified introductions to researchers, cancer center labs, diagnostic developers, and others. So if this isn't your space, you probably know oncologists and thought leaders who might be interested, and we'd love to connect and get an introduction if you are willing and able.